Spectroscopic publications, and he's currently the Director of Radiology at the University of Otago in New Zealand. He's won more than 10 awards for research, including awards from the Royal Society of New Zealand and the Royal Australian College of Radiologists, and he's lead investigator on over $12 million of New Zealand government grants. And Anthony has been a member of the scientific committee of the workshop in medical applications of spectroscopic X-ray detectors. And today he's going to give us a summary of the 6th workshop, not the 5th one. So,
yeah.
Anthony, please go ahead.
So I've got the wrong banner, so never mind. No one noticed it yesterday when I was giving the summary. So just to introduce myself, so thank you for having me here. It's always an honour to speak at CERN and in this auditorium. And one of the things I quite like about this post-COVID time, if I can say post-COVID, is we're actually better at sharing information. So I see there's several 150 people or more online at the moment. And I'm hoping that my friends in New Zealand are watching this rather than sitting around drinking beer and playing pool. But it is Friday night for them. So I suspect they're playing pool. So I've been asked by the workshop committee every time we've had the workshop to do a summary talk at the end. So listen to all the talks, summarize the field, and present back to the attendees what happens at the workshop. And so a couple of years ago, or a couple of workshops ago, we decided to also do a public talk around it so that everyone else can see what's going on. And I think that's really important. The workshop has been about bringing people together for knowledge sharing and to advance science. So this is quite an important thing. So the workshop actually happened in the council chambers next door over the last 4 days. And we'll go through the talks. Now, for me, my job's a clinician. I work in the hospital. I look at X-rays. I look at ultrasounds. My hobbies are physics and engineering. Some university gave me bits of paper saying that I can do those, but they're very much my hobby rather than my job. I'm also from New Zealand, which means I'm terrible at pronouncing European names, and I apologize for that right now. The talk also had to be written during the week. I had to listen to everyone's presentations, summarize them, and write a talk during a conference. And I'm a little bit naughty often when I'm at a conference. I tend to go to sleep in the odd talk and miss what happened. But in this particular workshop, I had to pay attention and try and learn one thing from every talk. I actually have a family member who has worked at universities, and he said when he was talking to undergraduate physics students, he reckoned he could get 1 point across every 5 minutes. When he was talking to the vice chancellor, he could get 1 point across every hour. So at this workshop, I was trying to get 1 point every 20 minutes. I've been associated with CERN and CMS and Medipac since about 2005. I think I officially became a collaborator about 2007. And so this talk and my view of the world actually is heavily influenced by that. It's about bringing knowledge together, getting it out to the people, and trying to improve healthcare in my instance. And a commercial disclaimer, I'm one of the founders of Mars Bioimaging. So the overview of the talk, I'm going to talk to you first about the people who came and the submissions and what we had. Then I'm going to talk about the context, so what's actually been happening in the last 5 or 10 years in medical imaging and what the drivers are. Then I've got a review of the talks. I've actually cut it down in the main presentation yesterday. I did everybody's talk, sort of one comment, as I say, per talk. This time I've reduced it so I can spend a little bit longer explaining to you what the main purposes were from just a few talks. And then I've got actually a reasonably long conclusion about what I learned, what this means for medicine, and what this means for the future. So starting with the participants. So that's the scientific committee. You can see it's headed by Michael. And then we've actually got all the major imaging vendors, Siemens, Philips, Canon, and GE. And then we've got a good friend or colleague from Hopkins. We've actually had it as an invitation-only conference. We did that because we wanted to bring experts in the field together, and we wanted active people. To actually attend the workshop, you have to be named on one of the papers. So that's kept the talks down to about the people down to about 130 who are all active in the field, which actually means there's been lots and lots of discussions. We had longer talks, about 25 minutes with 5 minutes of questions, and we had fairly long coffee breaks and social times. And one thing I actually enjoyed is that when people got together and they were socializing, about 2/3 of what they were talking about was work, which probably tells you about the passion and the feeling people have for their topic. So here we are. 113 registrants. This year, about 2/3 were from industry. And as I say, they were from all the major imaging vendors and some of the smaller vendors. We had 32 people from medical schools, and they were from reasonably well-known schools like MD Anderson, Mayo Clinic, Imperial College, not Imperial College, Royal Marsden. So that's actually exciting. We're bringing together really experts. And one of the more pleasing aspects, about 2/3 of the participants were returning. Those are people who have been before, found it value, and came back. So the context of the talk. Let's go right back to the beginning. We've been talking about what's called photon counting to get spectral information. Now, photon counting actually came out of CERN in the late 70s, early 80s, and Eric Heine was one of the key scientists. He's still at CERN. He actually turned up to the workshop, and they got an award in 2017 for their work on silicon strip detectors. That was the nidus of the scientific theory that led to all of this work that you're about to hear. Of course, Eric pointed out in this conference, it's actually photon processing these days. We don't just count the number of photons, we actually analyze them in quite clever ways, how many they're coming at, how they're interacting with the sensor layers. And I'll explain that as we go through. So that was Eric in the 80s. By the mid-90s, a collection of about 5 or 6 universities formed something called the Medipix collaborations. We're actually up to Medipix 4 now. And right back at the beginning, in the mid-90s, if you look at the original funding applications and proposals to form this collaboration, there was a view that if we use photon counting for X-rays, we should be able to influence medicine. So on that slide, there's 2 or 4 very, very visionary people. And it's taken a long time, but these days we're now affecting patients. Just a quick side issue into other types of technology. This is CT. So Hounsfield in the early 70s developed a CT scanner. Basically, you'd have an X-ray source and a bunch of detectors that rotate around an object, which often is called a patient, and you get a 3D picture. So this image over here on the right is a traditional CT using energy integrating detectors. So the more X-rays it hit it, the more it glows. And you can see that this person, this severe large vessel coming from the heart, the aorta comes back around the back of the chest, and this big grey blob here is their lung cancer. And so that was what state of the art was in CT imaging about 3 or 4 years ago. CT is pretty important. 300 million people a year get a CT around the world. I'll just show of hands, who here's had a CT? That's not bad, actually, only about a third or half the audience. If we were 10 or 15 years old, we might find it be 60 or 70%. Okay, it's a really common test. And so my view is if the scientists involved with this workshop have advanced the technology only a little bit, we've actually helped hundreds of millions of people each year. This is what I was meaning by spectral CT. So there's 2 views of the system. Here's an X-ray source and a single energy CT putting out a range of frequencies or wavelengths of X-rays. So typically we use from about 30 keV energy up to about 120. And then they go through the object, or attenuated, and it hit a grayscale detector, and you get a single unit, a scalar unit called a Hounsfield unit. What we've been doing, or what this collaboration has been doing, is trying to move that so that we have photon counting detectors or photon processing detectors to measure the energy of the X-rays, and then we get the spectral nature of it. So we can actually get the linear attenuation or the mass attenuation of the object we're looking at, and therefore work out what the material is. So in colloquial lay terms, we'd call that true color imaging. So when did the workshop begin? In 2011, there was actually a lot of skepticism outside the community. Around that time, I was visiting a lot of medical schools talking about the research that we were doing with Medipix and how it was going to change CT. And there was quite a lot of skepticism. I actually did one talk at a large US medical school where I had someone at the back literally telling me this wasn't possible. But the more common answer was, it's not worth doing. I think one thing I learned when I was a kid is when someone says something can't be done, what they're actually telling you is they don't know how to do it. Okay, and so people like Michael and Eric that I talked about earlier, they had the vision. They knew it was doable. And once one person's can do something, other people, of course, can learn how to do it. The other comment is not worth doing. That people have their fixed view of this is what I do today. things will only change a little bit if I had a new technology. And they're really saying the new uses are beyond their imagination. Okay, so at SpecXray way back then, we brought together a bunch of people. We brought the major vendors and about 70 or 80 people in the world working on this. At that time, we were showing with Medipix that we could do multiple contrast agents, so something that was impossible with traditional CT, so we could give iodine and barium in the same animal and distinguish it, so we could put them in different body spaces. Ewald, who was at Philips at the stage, he was imaging mice, and he was putting in contrast agents that were functionalized to go to atheromas, so the blood vessels that cause heart disease and stroke. So he's using the spectral nature to get molecular imaging or physiological imaging from the tissues. Jerry Aronson from Israel worked for GE. He built himself a prototype CT scanner. He was able to take a few slices through a human neck. It was really challenging. Having visited his site and talked to him, it was really squeezing the technology as far as you could, but proved you could actually do it in humans. So by bringing these groups together, we managed to say this is doable. And at the end of that talk, this came off my summary slide. In 2011, Jerry said, this may even be real. Clinicians want more information. The technology is almost there. And actually, there's going to be widespread benefit. So this workshop, by bringing the key people together, really brought experts who said, yeah, let's do it. Now, this was the first workshop we'd had in a while. I'm from New Zealand, so that's sort of down under our feet. And I hadn't been to Europe in 3.5 years, whereas I used to visit CERN twice a year. And so just returning for me was quite cool. So of course, the first night we had our traditional pizza dinner, and everyone enjoyed getting together. So it's been a long break, and a lot had happened in people's lives, and a lot had happened in people's technology. So it was good to get back. Clinical uses occurred, right? So this is a Siemens photon counting scanner that they launched at the end of last year. Neurologica, a small Boston company, actually bought out by Samsung, also have a scanner cleared for heads. And Canon are pretty much saying, look, it's here as well. So we've gone from a situation of 2010 or 11 where we had skeptics to actually having products on the market. And in fact, I'll show some more images of people and what we've been able to do. So I'll start with a review of talks. So we had Sahani, he's a radiologist from the US. He's fairly well known in radiology. He's been the previous chair of the American College of Radiology. I think he's the current chair of the Society of CT. And he talked about the benefits from imaging and how it affects patients. So he's not a technology person, he's a clinician who's driving towards things for the patients. And actually, these are the two slides he put up. And as the conference went on, I realized these were actually the two most important graphs from the entire conference. First one on the left, when I was training, I was at a medical school around here, junior doctor through this, but CT usage was going up by about 10 or 20% a year. It was massive, it was exploding. But around 2010 or 11, it dropped off. There was a distinct plateau. There was no benefit in doing more imaging. So what can we do? We're still not treating our patients properly. He also pointed out there's 4 ways we can help our patients. Patient safety, it's good not to hurt them. Diagnostic accuracy, this is what most people think of when they think of something to do with imaging or medicine. How do I do a better diagnosis? But he actually pointed out time to diagnosis or time saving is equally critical. So if you have a stroke, supposing, we'll think of my grandmother. She had strokes in the community. And that meant that she was sitting at home, arm became weak, and her husband called an ambulance. Okay, the longer it takes to get that person into hospital and start treatment, the worse the damage. And they tend to think in seconds for brain damage. For your heart, they talk in minutes of cardiac damage. For brain, they talk in seconds. So time to diagnosis is really important in patient outcome. So it's not that we can't diagnose stroke. The problem is when she was having strokes in the early 2000s in this area, the problem was to get her from the community into the hospital, make the diagnosis and start the treatment just took a long time. And the earlier we can drive that forward, the better. Cost savings. Cost savings is massive. We've got an exploding demand on healthcare all around the world. It's been completely disrupted by COVID. Most hospitals have backlogs of patients for and undiagnosed cancer and things sitting in the community. So we actually need to do things that improve cost. So first talk was by Cynthia McCulloch from Mayo Clinic. She showed lots of really, really pretty pictures of a Siemens scanner she's been using. So this is a dual contrast in a human. So 11 years after demonstrating that dual contrast can be done in mice, it's now being done in people. This is a brain scan at slightly higher resolution. And she pointed out the direct comparison with the traditional CT versus photon counting, where you get higher spatial resolutions and ability to see the contrast. There's actually a dural tail. So this is a meningioma, it's a benign tumor of the brain, and actually it's a cavernous one, in fact. And you can see here that dural tail is something the neurosurgeon needs. So you've got better diagnostic accuracy. From the 700 patients they've done, this is the number of benefits they saw for the patients. Reduced noise, improved quantitative performance, better iodine signal. Iodine is a contrast agent we give. It's a pharmaceutical we inject to make blood vessels show up. Being able to see that better helps with patients with diabetes and elderly patients. Reduced artifact, improved spatial resolution, lower radiation dose, and new applications. So this thing where in 2011 people said there's no benefit, Cynthia managed to demonstrate through her talk that they found sort of half a dozen to a dozen different areas of benefit. The next talk was from Philippe. Radiologist in Lyon, and he's been working with a Philips scanner. And again, they've been demonstrating benefits mostly around high spatial resolution that they can get with photon counting. Then I got to talk. We've taken a slightly different approach. We've been looking at putting our scanners into after hours practices. So this is a patient's wrist that's done on the CT, but we can see the tendons and the ligaments, stuff you'd normally get off an MR. Now, our goal is to say, well, photon counting allows us to use lower flux X-rays, low power X-ray tubes. That means you don't need specialized rooms. This particular scanner, I don't have a picture, or there's a picture of the scanner. We were able to put that in the corner of a radiographer's office, so we could actually take the technology out to the people. And so we've done about 70 patients there already. And I envision a series of scanners targeting specific body parts that we can push out into the community. And in fact, if you listen to the NHS, they put out a report, the UK NHS last year, saying they want to double the amount of radiology in the UK over the next 5 years, but they want it to be out of the hospitals. They want it to be where the patients are, because that improves the speed to diagnosis and reduce cost, which is what Sahani said in his first talk. GE have taken a very, very different approach. They've got detectors that are edge on silicon, so sitting vertically and X-rays come down. And they've got a scheme where they get multiple energies, and this is, I don't think this is real, I think this is simulated data, it might be real data, but they're working on it as well, and unfortunately they were unable to show any human images. We had a talk from a guy called Kareem from just outside of Toronto. He's developed a totally different technology, but still spectral imaging in medicine, where they have 3 layers of scintillators. So an ordinary chest X-ray, you get 3 colors in one exposure. And this is one of the patients he demonstrated to us. So there's a standard X-ray, digital radiography, anterior to posterior portable image that you might do on someone having a heart attack or in the emergency department with pneumonia. And he's separated out using the energies of soft tissues in the bone. And sitting right behind that clip, there's a pneumonia. Okay. Now I know that sometimes I make mistakes in radiology. I might well have missed that pneumonia on that single energy image, but with multi-energy, you wouldn't miss it. So that patient, they would have got a better diagnosis. We then went on, Canon showed some results. I talked about this being spectral multi-energy imaging. A lot of technologies out there at the moment are 2-bin. With Medipix, we're currently doing 5. And in fact, Canon's looking at 5 as well. And that just shows the massive noise reduction you get in having that spectral information. Raj Gupta, he's a neuroradiologist from Massachusetts General Hospital. He's one of these few people I've met who did his medical degree and works at Harvard, and then has a joint appointment from his physics degree at MIT. And he's, yeah, like a lot of overachievers, isn't he? But he's been working on portable scanners, And so this is a photon counting scanner that they've got at MGH, where they can take it into the intensive care unit, into the neonatal ICU unit, into the operating theatre, and into the interventional suite. And so again, that's using the new technology, the benefits of the technology, lower weight, lower power, to drive the imaging into places we previously couldn't. So actually in Christchurch in New Zealand, I work in the women's hospital 1 day a week. And for our babies that are in the neonatal ICU, we've got ultrasound, we've got X-ray. But if we want to do CT or MR, anaesthetists and the paediatricians and the nurses have to wheel an unconscious intubated baby to another department to take a picture. Much easier to be able to just move the scanner to the patient. This is some work from CHUV in Lausanne. They have one of our scanners up the road, and physicists, when they work with specimens, they like to show other physicists this is someone's toe that's been removed because they've got a huge amount of pain. And in fact, so they've looked at it and they use the spectral information to bring out a calcium map of the bone, but also they've been able to identify the crystals that cause gout. So this patient had so much pain in their toe, they preferred to have it amputated. And as she pointed out, this is where they see the CT scanners moving into the future. Dedicated CTs, single photon counting detectors and small voxels, and energy images and material images. So all of that technology is changing the way we use CT. This is again from the Philips machine. This is a coronary artery. Now, the reason I like this talk, I have a good friend called Latham, actually, who's a cardiac radiologist. And I remember about 5 years ago, maybe 7 or 8 years ago, we got a super fast scanner. It's called Flash from Siemens, and it goes fast. You can do stop motion cardiac imaging. And he called me over to his workstation and said, look at this beautiful image of the heart I've got. And then he pointed at that stent. So that's a little bit of metal that's been put in the blood vessel to keep the blood vessel open. He said, look at it, beautiful image of the heart, but where the stent is, where I want to look, I can't see a thing. And so with photon counting, that's the top image, you've got a clear view of the vessel wall through there, whereas the energy integrating detector, the older systems, this is what Latham would have been seeing, a blurry picture of the vessel that he couldn't diagnose. So despite the fact he had a pretty picture to show the other people, we couldn't actually use it to make a diagnosis. We had a nice trip out and walk around. Most of the people at the conference were from Europe and America. So I had to point out our oldest home building is 1835. And many people at the conference told me they had houses older than that. But we went up the lake to see the chateau. And actually, it was really good. I talked to a lot of other radiologists from around the world. There's Raj. He's from MGH, and Gatan is from Baylor School of Medicine. And they're talking about health systems that are stressed, huge waiting lists. You can't get people out of ED because there's no bed to put them in. You can't discharge them because there's no rehab in the community. So again, looking for ways to use technology to help our patients and to help our communities. Detectors showed some nice imaging. Detectors is from up the road at PSI, Paul Scherrer Institute, or it's been out there just beside me now, and comparing these are mice where they've injected contrast agents, 2 contrast agents, to look for osteosarcomas, that's that bit in the leg. And just, you can see the difference in energy integrating detectors where you've changed the X-ray energy by flicking it on and off. There's a multi-energy detector. You get a much better view of the cancer that mouse has in its leg. We then had a whole lot of technical talks on detector systems and packaging. So here you can see a detector. That's 4 units joined together with almost no gap. And so this talk was about moving from wire bonding, where the electronics comes off the side or the communication comes off the side, through to TSVs, through silicon vias, where you bring the signal out the back, and other methods for making these seamless detectors and allowing us to go to large areas. Oliver from Redland's in Canada, on Vancouver Island, they've been producing semiconductors for photon counting. So by high-Z semiconductor, we use something like cadmium zinc telluride. Because it's made of cadmium, zinc, and telluride. They're heavy atoms, they stop the X-rays quite well, a little bit like lead stops X-rays quite well. But as a crystal, it's a semiconductor like silicon. So you can stop the X-rays and you can conduct out the charge cloud and analyze the charge. And they've been building test units where they can look at the quality of the crystals that they're producing. And that sort of technology is what's required to take into medicine. It's not an experiment. You produce thousands and thousands of crystals, the same quality, and you measure the quality, and what goes into the hospitals is well understood. Matt Danielson from the Karolinska Institute in Northern Europe talked about these edge-on silicon systems. So there's the X-ray coming down into the silicon with multiple layers on it to measure the different energies. And he pointed out that with the silicon edge-on, you can thin it down and end up with a 1 micron spatial resolution, which would be fascinating to do. It's masses of data, but actually this enables something we call phase contrast. So X-rays, like light, are waves, and you can actually get interference patterns as they refract at tissue boundaries. And so the potential for photon counting or photon processing, as Eric likes to call it, is we can actually enable a new type of emerging called phase contrast. I like this talk. This was from Baha. There's an open source toolkit for MATLAB that allows you to simulate detector responses. And he looked at gallium arsenide. Remember those semiconductors I was talking about? It's another semiconductor that is used in photon counting. And the thing I really liked about his talk here, he's got a simulated data, the blue line, and his measured data, the red dotted line. Having simulated and measured data so close is amazing. Almost, yeah, I've never, well, yeah, it's pretty good. He's got a very, very nice simulation, hasn't he? You guys, a lot of you are physicists, you know how hard it is to get your simulations to match real data. Redden then talked about some of the problems in these semiconductors, and this is something that's going to come up a bit later on when I start talking about the new detectors that are coming out. If you have a crystal like this, say 2 millimeters thick and 300 micron pixels, you get an interaction. The photon comes down, interacts, and produces a charge cloud, and that drifts. But worse than that, some of these things, they'll hit a cadmium atom and you'll get fluorescence, K alpha and K beta, and they can come out symmetrically around the atom, and they'll travel a really long way, you know, up to 128, 168 micron through. And so if you've got pixels that are 300 micron, a third of a millimeter, this green line is how much of that charge or how much of that photon energy ends up being deposited in the neighbor, and that ruins your spectrum. Ideally, when you're measuring a spectrum, you want, if you put in a 60 keV beam, you want to measure 60 keV. Whereas here, they put in a 60 keV beam and you start measuring photons much lower down. And so compensating for that has been a real challenge, and I'll talk more about that later. Klaus from Philips, he talked about some of the mathematics they're doing for signal reconstruction. The first paper that I've ever seen, and I'm cited here, 1976, so 3 or 4 years after Hounsfield was publishing on CT, physicists were saying, it'd be quite good if we had energy information, and they started producing quite good mathematical models of it. Unfortunately, no one could actually build machines that did it, but I don't have a problem with that. The mathematicians and the physicists need to envision what's possible, and then we work out how we build a machine after. And through the last 40 years, there's been improvements in the way we solve these problems. Unfortunately, material identification, that's taking the spectrum and measure and deciding whether that's fat, water, calcium, or some other material, is a fairly difficult problem. It's almost singular matrix, it's ill-posed. And so his improvements, this is his noise, without regularization, there's heaps of noise. And by the time he's added his constraints, he's got the noise much, much lower. So much more accurate imaging. Scott's from Mayo Clinic, and he was talking about improvements in detectors. So there's 3 things, or he was mostly, first 2 points he was talking about is how do we reduce pileup. In medical imaging, we fire the X-rays off really fast. So we sort of have 10 to the 8, 10 to the 9 photons per second per square millimeter. Okay, so one photon's going to come down and hit the sensor, and we're trying to measure it. And the next photon's likely to arrive before we finish measuring it. We call that pileup. And so he was talking about schemes that people could do in the future to put into their electronics would actually measure that. He also talked about trying to measure the charge here. And remember I said that photon comes down and it spreads out either through electronic diffusion or through those secondary K alphas and K betas. He talked about having coincidence detection to measure that. Yode is from Phillips. He's actually on the scientific committee. He talked about the fact that we can now measure really low signals and we measure the energy. So normally in CT, we shine X-rays through and we measure what comes out the back and we only look at the shadows. But in fact, X-rays are scattering in all directions. And because we're so sensitive, we can measure down to individual photons in very low noise environments. You've actually got that scatter information that's measurable. And so here he's done a reconstruction without scatter correction and one where he's used the photon counting ability to do scatter correction. I think that's a really nice demonstration that actually new detectors, new technology allows us to bring in new information. Now, this is actually something I learned from undergrad physics. One person's noise, in this case scatter, is another person's signal. So by what everyone else is saying, oh, I've got too much scatter, it's too much noise, he's saying, I want that scatter, I'm going to use it for reconstructing my images. Then Pierre talked about reconstruction. There's two parts to the way we take our raw data and turn it into an image. The first part, or that can be done in any order, is you have what are called sinograms. So that's the raw projection data. So it's around the patient, and then you turn that into a 3D volume. That's called CT reconstruction. And the second part is to take the spectral nature, which is attenuation at different energies, and turn that into materials like fat, water, calcium, and iodine. And so that's a two-step problem. And there's been debate in the literature for a long time whether you start with the spatial reconstruction and do the material reconstruction, whether you should do the material reconstruction, do the spatial. And different groups have different preferences. Most believe their way's right. I tend to have always thought they're both wrong. It's the same process, it's the same physics. And so a one-step approach where your algorithm does both reconstruction and material identification is clearly the right way forward. And so Pierre put forward a really nice method. There's a few other methods in the literature. Then we had talks on contrast agent and molecular imaging. So most of what people think of an X-ray or an MR is what I might call anatomy or morphology. You get the shape of an organ, you see that a cancer is this big, right? But you don't know what cells are in there. You don't know what physiological processes are going on. What surface antigens do those cells have? So Ketan from Baylor, it's actually the same image from earlier. He's taken contrast agents where he takes a bilipid layer, pegylates them. This is all biochemistry that I never learned at university. Puts an iodine and gadolinium and then injects into the animal. And you can see he's got different surface markers. So one of them will go to different parts of the tumor than the other. So that's allowing molecular information or functional information directly from the animal. And so in fact here, it's that tumor, that's that green blush through there, has whatever surface antigen that is. Okay, but that's molecular imaging, chemistry-based imaging. I then did a talk on behalf of one of my colleagues who couldn't unfortunately travel due to COVID. And she's been collaborating with groups from around the world. So here she worked with University of Illinois with hafnium-based nanoparticles looking for microfractures. So that's a human finger. We didn't put a picture of the finger in, but it's been excised from the person. And the nanoparticle targets where there's been bone damage. This is work she's done with the University of Madrid, where they've taken a contrast agent that's red and identified where the TB granuloma are in the lungs. This is work done with University of Pennsylvania, where we've looked at atheromas. So in this image, the pink is the ear tissue interface, which is why you can see pink on the outside of the animal. So the pink there is the lungs, so all the surfaces of the lungs. And then we've injected in monocytes, a cell line that occurs with inflammation. And those monocytes had previously been fed gold. And so here, coming up over the bronchi, there's the aorta, that big blood vessel in the heart, and those monocytes, we track them into the atheroma. So again, it's cellular-based imaging done with David Commode. And this is gut imaging. So again, a pegylated nanoparticle that has gone to the mucosa in the gut of an animal, and we've been able to identify where that functional agent's gone. So spectral imaging is allowing us to go from anatomy through to imaging biochemistry. We then had a few talks on simulations and materials. So Stefan talked about several things. The first thing he talked about was this charge sharing problem and how difficult it is. So he's modeling this. And then whoopsie daisy. And then he talked about the pileup problem. So there's two photons arriving very close together in time and the difficulty in separating them. And so he modeled detectors and mathematics that would actually allow you to reconstruct without these two problems. Okay. Vico from Germany talked about the breast CT they've been doing. So this is, if someone has a suspected breast cancer, they might have been picked up on screening 2-view mammography. But actually, you then need to work out what sort of cancer it is and what sort of treatment there will be, whether there's secondary deposits. And so they've got a CT scanner. The woman lies prone, and what's the words they use? They put a pendulous breast through that hole. But that sort of hangs down, and then you rotate around and take your pictures. And he looked at the difference. You need to have 100 micron voxels to see cancers in the breast, or to see the speculations or the parts of the cancer. And he looked at that charge sharing phenomenon, that spreading out between pixels. And if you don't correct for it, you get a nice CT image, but your ability to remove the contrast agents and look at the iodine only just becomes really bad. You've lost that spectral information. Whereas if you can correct for that charge sharing, you get beautiful images of the iodine in the soft tissue. This is all going to come up when we talk about the latest detectors. Juha's from Finland. I've known him for a long time, and he was talking about gallium arsenide, so that new semiconductor for detectors. And he's, with several local partners in Western Europe, developed gallium arsenide. They put in some chromium, I think they diffuse it through from both sides, and can produce really nice wafers that allow us to take very, very high resolution images. So that's another detector material beyond the cadmium zinc telluride I was talking about before. Edgar's from Siemens, and he actually really kindly told us about the detectors they've got in that Siemens machine. It had to be 4-side buttable, so they can produce strips of it. So they've gone through the TSVs. They've got 4 energy bins, and he told us about the count rates and linearity, and their pulse, their spectrum measurements that they get off their machine. So that was quite a nice technical talk about the inside of this commercial scanner. Peter gave a really nice explanation of designing electronics to compensate for that charge sharing. So there's your photon coming in, illuminating several pixels, and the charge is drained out, and you've got that charge sharing phenomenon. And as he said, there's many detectors. The one I use is SpecXray, designed by CERN and partners, where we actually correct that charge sharing. So I talked earlier, there were lots of different people saying, you've got to do this, you've got to do this. There are now, well, I think SpecXray was, certain SpecXray was the first to actually have charge sharing correction, but everyone else seems to think it's valuable now too. And then the final talk of the workshop was from Veros, and he was talking about Medipix 4. This is just back from the foundry. I don't think they've diced it yet, or they're just in the process of dicing it and mounting it. It's not actually electrically tested. But within the ASIC, they built in pileup correction. So for all of those photons that are coming too quickly, it works at which is those pulses that are together. It measures them and works out what's happening. So that allows us to go to much, much higher count rates. Of course, in Medipix 4, they've got the charge sharing correction that Medipix 3 had in it, and they've got a 4-side buttable detector. So this is where I show my bias. I believe Medipix 4 is actually the most advanced ASIC for CT and medical imaging that's around. Copes with high flux, high energy resolution, and the extendable active area. So all of the problems that we were hearing about for 2 days in detectors and reconstructions are addressed by this detector. It's not perfect yet, but it's getting there. And finally, we had an invited talk on antimatter from the LHCb. That was actually really fun. As I said at the beginning, physics is my hobby. And so it was really nice to actually hear straight from the I was about to say the horse's mouth, that's unfair, from the knowledgeable person's mouth about what work's going on here. So in conclusion, let's talk about this for a moment actually. In 2011, when we started this workshop, there was a question, would any of this work? By 2022, there's wide acceptance of the technology feasibility. There's a bunch of people doing patients now, either in clinical trials or approved for use, sort of 3 or 4 different groups. My group particularly is focused on taking our imaging out to the community. There's wide acceptance of the clinical utility across a range of imaging problems. So I don't think we're there yet. I mean, the machines are only just starting to be used on people in the last 6 to 12 months. I suspect there's a decade of new uses to come. And I think it will completely replace traditional CT in the same way that cathode ray tubes were replaced by flat panels and TVs. I think the same will happen. And just like when we went from cathode ray tubes to flat panels, we developed a whole new range of applications for these technologies. So flat panels, of course, enabled laptops and cell phones. If you still had CRTs, I wouldn't want to lug around with a cell phone in my pocket. This is Stefan. So Stefan heads one of the Siemens research labs in Germany. I think the one just outside Nuremberg. It's quite a big lab. And actually in the 10, 12 years he's been on the committee, he's gone from an engineering researcher to head of the lab. And that's sort of an interesting career progression. And when me and Michael and him were talking at our conference dinner, he actually said some really nice things. He said, basically, when we brought the community together, there were a bunch of scientists at both industry and academia who believed this was possible. But until we actually got the community together and produced enough kind of mass or momentum, we couldn't really move the field. And he believes that the SpecXray workshop has been important for that. And he also said some very nice things about the Medipix collaboration, basically proving it's possible. And of course, if you're an engineer, once you know something's possible, you can find other ways to do it. But the first person to do it is bloody hard. So he said that over conversations, and then he wrote this little public statement that we can put up. But the final part of my talk, of course, is this. This is what we're trying to solve. CT has not been significantly improved in about 10 years. Usage is pretty much flat. And I think with Spectra, we're going to have new applications and new places to use it. So as I said, I think this is almost the most important graph of the conference. And we actually have to address several things. Diagnostic accuracy. Okay, can we see and make more diagnosis? Time saving. So there was myself and the Mass General radiologist saying, we've got to take the scanners to the patients. We can't have the patients come to the scanners. And cost savings. This drives usage, right? Unfortunately, some of the most wealthy health systems in the world, like the US health system, spends, I think, 14 or 16% of GDP on healthcare, and they've got falling life expectancy. We've just got to address some of those problems. So in the spirit of CERN and knowledge sharing, what we're actually trying to do in the conference is bring everyone together, share our knowledge, take it out to the communities, and improve the lives of our friends, our families, and our patients. So thank you, and we'll be doing this again in 2 years. Happy to take questions.
So thanks, Anthony, for the nice presentation. I'll take questions in the room first. Okay, maybe I'll start. So basically, you said we're in the clinic now. So after 10 years, we got there. Why should we keep going?
That was a really good debate that we had as a scientific committee on the first day. And then I actually went and surveyed people or questioned people. And actually, Scott, in one of his talks, he played the sound of a modem from the 90s and said, that's where we're at, right? We've just started to get the technology there. I think there's going to be 10, 20, 30 years more technology development. We can vastly improve the quality of the detectors. We can vastly improve the quality of our sensor layers. And our reconstruction, I think, has probably got 40 or 50 years of mathematics to go. So by bringing the experts together, because these are very disparate fields, CT reconstruction versus microelectronics versus radiologists, unless you can bring those experts together and see what everyone else's problems are, it's actually very hard to advance the field if everyone's working in isolation. So the conclusion from all the attendees is we'd like to get together again.
Please.
Just a curiosity. How does this affect the cost of a CT machine?
That's a really good point, because
In fact, you're talking about overall reduction of costs, things like that, but seen from the patient, is the CT going to cost more than it does today?
That's a really good point. So one thing that's really clear is big manufacturers are bringing in machines at the top end of the market, you know, a few $1 million a machine. And I think that's going to be really challenging because most health systems actually can't afford the systems they've got already. But I think the technology is more of an enabler. So photon counting works really well at low flux, low X-ray power. So in our scanners, we have a $10,000 X-ray tube. In a traditional CT scanner, you have a $200,000 X-ray tube. The Siemens guys told us yesterday their photon counting system for speed has 2 X-ray tubes at right angles. So just in your source, you've got 400,000. But in fact, with photon counting, because you can go much lower power if you want to, produce cheap machines, air-cooled, lower dose. So one of the big savings we have, I was talking about it with Raj from MGH, when they put in a new CT scanner, they spend as much on refitting the room as they do on the scanner, which is, you know, $1 million to refit in Boston. Whereas the scanner we've got in Christchurch, we move, because it's low X-ray power, we put it in the office of the radiographer. We could train the radiographer in 4 hours because all they need to do is put the start and end position. You don't need to have all these fancy things about what power you're using, what energy you're using. You just do the whole lot. It's just an anatomical thing. So I think actually it has the potential to drop the cost significantly. Again, I'll hark back to that cathode ray tube versus flat panel CT. You change the technology, you actually produce the opportunity for reduced cost and new uses of it. And so you can change the way it's used. Whereas I think the big companies have come out with top-end scanners that are expensive. And I think actually that's almost missing the major benefits of photon counting, that we can do it in places that we couldn't before. So I was talking at morning tea outside with the guy from Baylor, and he does a lot of head imaging. He says he wants scanners in the ambulances. When, like I talked about my grandmother's stroke, the scan should be done in the ambulance on a small portable light system, low X-ray power, because you can't have an X-ray source sort of cruising through your city without lead shielding around it. So the doctors know by the time the patient's got to the hospital, that person's having thrombolysis. We're going to inject them. That person's going to the interventional suite. We're going to put a catheter and pull the clot out. So there's a cost issue, but it's not just the cost of imaging, it's the cost of treatment. And so by improving where we do the imaging, how we do the imaging, we can actually reduce the cost of the health system.
I think it's also the aspect of, in some cases, replacing MRI, and in some cases, replacing PET.
Yes. Yeah. So I'll actually tell another story that was from my talk. So we had two patients, one of which happened to be a family member. He fell off his bike, hurt his wrist. He did what most people do. They went to the local community doctor. They had X-rays there, but that's the only imaging they had. So he got referred to ultrasound the next day because they could do that there. Then he got referred to the orthopedic surgeon. Then he got referred for an MRI. Then he got referred back to the surgeon. Then he got treatment. It took 11 days, 12 hours of his parents' time driving him around to get these things. So the cost of that diagnosis was massive. Whereas we had another patient in the same city from part of the trial. I remember she fell off her bike and hurt her wrist, and she happened to go to where we had an after hours at one of our scanners at the after hours clinic, and got scanned then and there, and it made the diagnosis of a broken wrist. And instead of having that pathway where lots of different imaging tests were done, took 11 days. It was an unexpected finding. We didn't think she was going to have it. We called up the orthopedic surgeon. She was treated that day. So the total cost of the diagnosis went down because you've done it earlier. We eliminated an MRI, we eliminated an ultrasound, and started the treatment earlier. So the cost of care can drop significantly with this new technology.
Christian, please.
You showed fantastic CT scans where one could see organs, blood vessels, and so on, already colored. This color or this coloration, is it done by the machine, or does the clinician have to do it manually afterwards?
Right, yes, yeah, so I did sort of skip over that. So what we do with Spectral is it's X-ray color, and then we convert that to materials. So we convert it to iodine, gadolinium, fat, water, calcium, and then the doctors tend to like it shown as multiple black and white channels. So that's the water channel, that's the iodine channel. For conferences, people tend to overlay them with colors. They'll make the fat white or the bone white, the iodine red, the gadolinium green. So those are just colors for display. In fact, what you've got is the partial density of each material. So we should actually use black and white images with units of grams per cubic centimeter.
Any other questions in the room? Yes, please.
Hello, very nice talk. Thank you very much. I have a question about contrast agents. What do you think as a radiologist? Like, is there a place for new contrast agent? Is there need for new contrast agent beside iodine and barium? And do you think that it's feasible that this new contrast agent will get approved in the future? What is the field right now?
Okay, so that's a really good question about contrast agents. I'm going to divide contrast agents into two types. Non-functional ones, which is a standard iodine or gadolinium that used in the hospital now. They have trade names like Ultravist and then functionalized contrast agents. So starting with an unfunctionalized one, we already use them in a hospital. They have two problems. One, they cost money, and two, they're toxic to the kidneys. So anyone over 60, you have to do a blood test before you give that contrast agent. Because we're better at seeing the iodine, pretty much everyone in this conference agreed that we could halve the dose of iodine. And that's massive. That protects the kidneys. That's actually quite a lot safer. And actually, I think it's about 20 or 30 US dollars per patient to give iodine, just that ordinary iodine. And so you're dropping the cost of the study by 20 bucks just by halving the iodine. So for non-functionalized contrast agents, it's cheaper and safer. The functionalized agents, this is where it gets really interesting. I showed a few pictures from our group and from Baylor. Molecular imaging is currently done on PET, like PET CT, PET MRI. That means you need a cyclotron. It means you need a facility with concrete walls. It means you have to have one-way passage of your patient. It means you need to have a radiochemist, and you need to have typically 2 or 3 technicians. So in New Zealand, only about half the patients with cancer end up with a PET CT because it's expensive and only available in tertiary hospitals. And if you live in Greymouth, you're a 300-kilometer drive from a tertiary hospital. It just doesn't happen. With the new molecular agents that are coming out, where you can functionalize something, you don't have radioisotopes. So they tend to be stable. You can put them in the fridge. That means that if the patient needs it at 2 a.m., the technician can take it out of the fridge and inject it without the need for a radiochemist. You don't have to have a cyclotron, and you can have multiple agents. So you might have in your fridge one for looking at blood vessels, one for looking at amyloid deposits in brain disease. And you can actually then have a range of them. So I think there's a challenge in getting those new pharmaceuticals into the market. But now that there's imaging devices, it's going to be easier. But I think the benefits of getting those molecular agents through into the hospital are huge. It's going to provide better access to patients. It's going to be safer and cheaper.
Any more questions in the room? Burkhard?
Yes, I have been looking again at the plot you had shown before the last one, which you said is an important one for this workshop you had. Now, the stagnation you show there is over a period of 10 years at least. And if I understood well, the message you gave also is that the methods you described have been publicized mainly over the same period. So somehow it looks a bit disappointing.
Well, what's happened, this period here, I'll go through that. This is when we got so-called multi-slice. So we went for faster and faster acquisitions and higher spatial resolution. And what that enabled is more and more applications. You kind of got to a level of speed or resolution where you could suddenly do the next problem. And then we got to a point where through here, we've had faster and faster scans, higher and higher spatial resolution, but it hasn't affected diagnosis. So the manufacturers have been putting out really fast machines, the whole body in half a second at half millimeter resolution, but it hasn't changed what you can diagnose. So that's when the stagnation occurred. We've only just in the last 6 months got patient imaging with photon counting, hence my hope of the arrow going up. So that stagnation actually represents the maturity of that technology, right? This is the pre-phase, and that's where photon counting is. I'm hoping to do that. And I think it's I think it's not just the diagnostic accuracy. I think it's this time to diagnosis and cost savings that actually might drive the adoption.
Thank you. So we look forward to see what the situation in 2 years.
Yeah, it's going to be exciting actually.
Are there any questions on Zoom? Yeah, in the chat. The first one is, it seems it's time to think on how to add this to gantry as part of dynamic CT.
Sorry, can you say that again?
It's time to add this to the gantry as part of dynamic CT.
Adding this to dynamic CT. So I think by dynamic CT, there's two or three places we use dynamic CT. One of them is the interventional suite, where we have a C-arm that rotates around and you're putting catheters into the patient. It's clearly time to do that because you're injecting contrast agents. By the time you're halfway through a procedure, you might have injected 4 times and you're wondering what's the latest injection, what's the first one. And so if you've got multiple agents, you can actually start telling what you're doing. The other thing that you've got is catheters that you've got in the patient. And if you label the tip of one catheter with gold and the tip of another catheter with something else, it's easier to see what you've done. So I think, yes, Spectral will be good there, but they're large area detectors, sort of 25-inch detectors. And that will be possible with the 4-side Butterwell, but it's going to be the other type of dynamic CT we do a biopsy. So that's traditional CT where you're putting, say, doing a lung biopsy. Yes, it'll be helpful there too.
Another question is, what is the biggest advantage of colour X-ray CT over MRI and traditional CT?
Over MRI is access. MRIs have relatively high field magnets, 2 and 3 Tesla magnets. They require, in most countries, I was talking to other radiologists, 3 technicians to run them, 2 techs and 1 nurse, and they're slow imaging. Okay, and they're therefore expensive. So you might be putting someone on the scanner for 40 minutes. It's a $3 million machine in a $2 million room, and if someone's got metal in them, they can't go in. So if they've got a pacemaker, you can't put them in the MRI. If they've had a knee replacement, which is probably why they're having an MRI, because their knee replacement's sore. Of course, the metal totally screws the background magnetic field. You can't see a thing. One of our collaborators in Hong Kong has a beautiful image of an X-ray and an MRI of someone's knee where they can't see a thing. And then they've taken, well, actually the knee taken out of the person, put it in a spectral scanner, and they can see everything. So they're safer, cheaper, and better diagnosis.
Simon Connell on Zoom has a question.
Yeah, hi, thank you very much. I'm in South Africa. I would like to have clarification on 2 points. I think I heard that you said if you knew the color of the photon or the energy, then you had almost like a greater penetration, or you could get by with lower energy photons. That's the one point. I'd like just an explanation of that a little bit. And the other one was, it seemed to be that the flux required for sufficient image statistics could be lower. You could get a better image with a lower flux. I think that's what I understood, but could you please clarify? Thank you.
Yeah, okay. So there's a couple of reasons that you can get away with lower dose and lower flux. First one is you get rid of the electronic noise. So you can actually measure down to, well, photon counting, so you can measure a photon a minute if you wanted to. So the traditional energy integrated detectors have to have a sufficient number of photons hitting them to get a signal. Counting almost has the opposite problem. It works really well at low flux and poorly at high flux. So you shift down the dynamic range that you're targeting. The second reason is by having higher frequency of pixelation in the detector, you get better transfer of the signal into the scanner. So actually, Norman Pelk from Stanford's got a really nice couple of papers to put about 5 years ago about higher sampling frequency and how that helps with reconstruction. And the third aspect is actually simply to do with the mechanics of the old integrated detectors. They were crystals, cesium iodide, I think, and each pixel would have a little boundary between it, not the non-scintillator, otherwise the light in one pixel would diffuse into the neighbor. And as those pixels got smaller and smaller and higher resolution. About a quarter of the detector ended up being the barrier between the pixels. So you've got about 25% dead space, and that goes. So there's 3 reasons that you're going to be able to lower the dose.
Thank you very much.
Are there any advantages of in the cardiac field, especially compared to cardiac MRI?
Yeah, absolutely. So in this talk, I showed around the stents, the cardiac stents that are put in for the narrow blood vessels. You can actually see inside them. The other advantages is characterization of atheromas. So most imaging of atheromas is done basically looking at the lumen and narrowing it. So if you're doing ultrasound, there's a NASIC criteria that tell you how narrow the vessel is and therefore whether you need to treat. And the characterization of the atheroma, you've got various parts. The key one is whether there's an unstable plaque, and it's a little bit unsure what causes instability, which one's going to rupture and cause a heart attack. But it's thought to be things like fat content or inside the plaque that may be part of it. The stuff I did with David Commode there, where we had a mouse, we were looking at the monocytes, that's the inflammatory component in a plaque that he's measuring. We actually published some papers a few years ago where we looked at iron contents and microhemorrhage within a plaque. So you've actually got better depiction of stability. Your question was specifically around MR. MR can pick up the fat in the atheromas, but actually MR is not very good at high resolution compared to photon counting. You know, there were people here talking about getting down to 50 microns. Most of the imaging on photon counting now sits between 90 micron and 300, and MRIs tend not to go down to that resolution.
Thank you. One last question from the room. I think we're about running out of time. Oh yeah, Manu.
Anthony, a curiosity, because we're speaking about the detector side, but to implement the full CT, then there's all the rest. Is there any other limiting factor or areas that would need improvement to get to the photon counting CT of your dreams? We think about the X-ray source. Is there anything on that side?
So I kind of break a CT scanner up into 4 parts: gantry, tube, detector, and mathematics, right? In parallel to all this work in detectors, there's been quite a lot of work on the sources. The sources in, you know, like a standard CT like the G Revolution are huge. You know, they weigh 20, 30 kilograms, 20 amps power input, sort of thing. But in fact, with photon counting, you go to lower power, and that enables new sorts of sources. So one of the sorts of sources I've seen is a distributed source. So they're low power, they're sort of milliamps, but you can have a line of them and you switch them on one after the other. And that means you can have a ring of sources and a ring of detectors, and that means no moving parts. And so that becomes much, much easier. Not to have slip rings. Your cooling's better because you fire this one off and then you fire the one on the other side. So the actual duty cycle for each source is much lower, cooling's easier. And actually that improves the mathematics of the reconstruction as well. So one of the problems you have with a traditional CT, you've got a trajectory where you're spiraling around the patient. If you're firing them off almost in a random way, you get rid of a lot of the artifacts that come from the fact that you're spiraling. And so that improves. And then finally, the lighter, I'm not finding because I'm only halfway through here, with the lighter mechanics, no moving parts, your gantry changes dramatically. So even Raj Gupta's portable CT scanner was still kind of 500 kg on wheels. If you can reduce the X-ray power and all sorts of things, you've got a CT that might go in an ambulance. So no moving parts, low power, you can literally put in an ambulance. And the final part is of course mathematics. It's not an easy problem to solve, the reconstruction optimally and the material decomposition. And I've been participating, but more watching scientists on that for 20 years, and it's clear that there's a long way to go. You know, everyone's getting better and better at it. You know, the CNN convolutional neural network-based reconstructions that halve the amount of information you need and stuff are great. So that's going to go on for another 30 years as well. So I foresee all parts of the system actually having a very long lifetime of research.
Maybe just another point on that is there is some other work, but not in medical applications, but inside the Medipace collaboration, where people in Prague have a detector unit which is carried by a robot arm and a a source on a robot arm as well. And those collaborating robots can basically scan any object. And so there you see that this new technology where you basically don't have the same requirements in power and cooling and all that kind of stuff. I think those kind of combination of our detectors with new technologies will bring revolutions we haven't even thought about yet, I think.
Yeah. You change one part of the core part of the system, like the detector, everything else can change and need to be rethought about. In fact, one of the almost disappointing things is how people have said, oh, well, we've changed the detector, we just had to squeeze it into the old system, rather than saying, we've got a new way of measuring stuff, let's design a new system that matches the detector.
Okay, so
I have a question about, is there triple energy subtraction capabilities available now?
You can do triple energy subtraction, but we tend to actually measure at least 5 energies. And so it's not so much that we're subtracting, because I think you're thinking of dual energy subtraction, which was the older technique. When you're measuring 5 energies, you're not actually just doing subtraction, you're doing a basis transform.
Yes, you identify the composition of each voxel.
Yes.
From that you make the energy.
Yes. Yeah.
Okay, so let's thank Anthony again. Thank you.